This is a journal club that we've been holding at Johns Hopkins for about 4 years. And during the COVID-19 shutdown, we decided to take it virtual. So this is, I believe, our 5th session. We've had a wonderful response from the community. So thanks to all of you who have joined and contributed and made the discussion very lively in previous events. So I'm happy today to present our speaker, our discussion leader, Liang Dong. He is an MD. He is currently working with Ken Pienta's group at Johns Hopkins. So the Pienta group has some amazing research that they're doing on cancer in the Brady Urological Institute here at Johns Hopkins University. And the paper that's going to be presented today is about how we could potentially use PD-L1 on extracellular vesicles to monitor melanoma. So this is a publication in the Journal of Extracellular Vesicles. And I'm going to now turn the microphone over to Liang. Thank
you very much, Ken. So can everybody see my screen right now? It's the slides mode, right?
Yeah.
Cool. Yeah, so it's my great pleasure to be a speaker today, and welcome to the EV Journal Club. So I guess since most of you may not know me, and Ken just gave an excellent introduction, and I'm not from Dr. Whitworth's lab, but I do work for another Ken in the same campus in Johns Hopkins. So currently I'm working as a postdoc in Department of Urology, and I'm personally a urologist. So how can I do next? Okay, so this is the paper I'm going to present today, and it's published early this year on the Journal of EV and from a French group. So the title is Tracking the Evolution of Circulating Exosomal PD-L1 to Monitor Melanoma Patients. So to be honest, as a non-native speaker, I'm a little bit confused in the beginning as the evolution, with the evolution, the title. So I think, according to my understanding, it's something like a dynamic change of levels. So as an introduction, I'm a urologist, not a medical oncologist, not a immunologist. So you guys may be interested, like, why I choose this paper. So I chose this paper for several reasons. First, as I routinely check the website of JEV, yes, I'm a big fan of that journal, and I found this paper recently published actually had one of the highest number of viewed, so it's very, very popular. And second, PD-L1 actually, and not just PD-L1, also other immune checkpoint inhibitor is not just for melanoma one disease. It's almost a pan-cancer. It's actually worked for many other cancer types, including, not prostate, including bladder cancer and kidney cancer in my field. And last but not least, it's a clinical study. I won't say it's a pure clinical study, but a lot of clinical work. So I'm more interested. And when I see this title, I think, wow, that's something I was planning to do for a while. And that's why I choose to present this. And also, a lot of things I also try to do, and there are a lot of good try and data. We can learn a lot of things in other field, other diseases. So first, I do have a statement about nomenclature. So just want to be concordance with my self-guidelines. The term extracellular vesicle or EVs will be used in this presentation instead of exosome or exosomal. Just want to be more accurate and precise. So I know the ISEF, especially Dr. Whitworth and a lot of other experts spend a lot of efforts trying to establish guidelines and protocols in this field. I do appreciate it. And however, I will not change any terms in the figure and table, just as respect to the authors. So first, in the beginning, I put a few slides about the background. Though I know many of you are already very familiar with these terms. So just want to give a very brief introduction. So the PD-L1 also stands for programmed death ligand 1. So it is a membrane-bound ligand found on the cell surface of many cell types, not just cancer, that is upregulated in the setting of inflammation and or a number of oncogenic lesions. It binds the PD-1 receptor on immune T cells, leading to the suppressed antigen-driven activation of T cells. And this mechanism normally keeps inflammatory responses in check. However, tumor cells can actually use this mechanism to evade immune destruction. And actually, the whole picture for the immune checkpoint inhibitor is very complicated. And just want to make the long story short, the take-home message for this paper, I chose these two images. I think this is enough. So what we need to remember and know is that PD-L1 expressed on tumor cell, once it binds to PD-1 expressed on T cell, then the T cell will not kill the cancer cell. However, if we block the PD-L1 or PD-1, and then it allows the T cells to kill the cancer cell again. And this is the basic mechanism. And this actually has been applied to the clinical setting. And so immune checkpoint inhibitor have shown effectiveness against a large number of cancer types. This response includes durable remission in many patients who actually have previously failed multiple other treatments. And because of this like dramatic change and contribution, so two scientists who did fundamental work in this field were awarded with the Nobel Prize in 2018. And I believe everybody knows this. So on the left side, I just posted a figure from a recently published paper reporting the results from a phase 1 trial of Atezol, which is a PD-L1 inhibitor for metastatic melanoma patients. And as you can see here, in these late stage cancer patients, about 30% of them have a response of different level. And look at the figure. The y-axis it indicates to the percentage like change in tumor burden, and the minus like 40% or 80% means like tumor burden largely decreased. And the x-axis indicates the time after the beginning of the study. So this is actually really amazing to me, because it's not just 30% of the patient can live longer, but also, as you can see here, the response is not like immediate response, actually can last for a while. And just like other like PD-1 inhibitor and the PD-L1 inhibitor, so all these research, if you just look at the 30%, the number, you may think like, oh, not all the patient has a response, but actually these patients, all of them, years ago, they don't have any other options. So this is really like great improvement. So however, look back to the number like 30%. So overall, like all these studies, like only 10% to 30% of patients respond to anti-PD-L1 or PD-1 therapy. So there is actually an urgent need to validate a biomarker allowing selection of patients who might benefit from these treatments. So in the beginning, it was expected that the PD-L1 should be locally expressed in tumor tissue for these drugs to have an effect. This makes sense because we want to like, the target is PD-L1, PD-L1, they should be actually expressed in the tumor, right? However, this may not be true because PD-L1, the tissue stained PD-L1 level remains to be an imperfect predictor of these kind of drugs. So look at the figure on the left side. So I pulled this figure from a review paper published 2015, a little bit early. As you can see here, there are 4 drugs here. The first 2 are anti-PD-1 drugs, and the next 2, they don't have a name at that time, but now they do have a name. They are anti-PD-L1 drugs. So the blue color, the blue bar are the all patient, their response rate, and the red bar is the patient with a positive PD-L1 staining, and the green bar indicates the patient with a negative PD-L1 staining. So the review actually gathered all the data, and we know that in a lot of research, tissue stained PD-L1 actually can tell us, can predict a response, but not all of them. As you can see here in some of the study here, and there. So the PD-L1 staining actually cannot predict the response. And another fact is like in almost all of these studies, there are many patients with low to like even no detectable PD-L1 expression who still like actually experience like a successful like treatment. So we actually missed a lot of patients if we only use this as a indicator. So there are a lot of like research and hypotheses. So it is possibly due to the heterogeneity of the PD-L1 expression in the tumor tissue. So the heterogeneity could be like spatial, so we didn't biopsy the right place, or it could be actually changed like through the time. And also it's possible there are some other unknown biology actually exist and play a role. In recent years, scientists start to think about what kind of role EV may play in such a very important like biology. So EVs, extracellular vesicles, are lipid bilayer encapsulated particles that can be released by all living cells into the extracellular space. And EVs carry a variety of molecular cargoes, including proteins, metabolites, and nucleic acids, DNA, RNA including like mRNA or other like non-coding RNAs, and can transfer their cargoes to target cells either by locally as a paracrine signaling or by traveling to a distal body site. So through these years, EV, like the society like agreed EV play a very important role in cell-to-cell communication and serve as promising biomarkers in liquid biopsy. So here is a figure I took from one of my review paper published last year. And as you guys can see, some biology already need to be revised because our field is really changing so fast. However, this just give a very brief summary of like the EV biology from the biogenesis. So briefly, there are two mechanisms. So the EV can be generated either by the inward budding of the cell membrane and go through like an endosome pathway and pack the cargoes and eventually be released. And we call them like exosomes, or by directly outward budding to form like exosomes. And then like in the as a transportation, so the lipid bilayer actually provided very safe shelf, and then to protect those cargoes. That's why like we can easily find like EVs in any kind of like biofluids and take them as like a biomarker. And then when the EV travel to the target cell or receive cell, there are multiple ways for EV to play a function. I won't go too deep because that's not the point for today. So in the past 2 years, there are two very important papers published, one on Nature, one on Cell, focusing on the role of EV or the PD-L1 expressed on EV. So now we know EV carried PD-L1 actually contribute to immunosuppression and is associated with anti-PD-1 response. So there are a lot of huge amount of information in these two papers. I just selected some of them, which is related to this paper, and are listed on the right side. So first, these papers found PD-L1 were found widely expressed on cancer cell-derived EVs. They do exist. And second, EV PD-L1 actually do have a function. They can suppress T cell activation, not only within the tissue, but also in a distal site, remotely. And the third, PD-L1 negative cancer cells can secrete a large amount of PD-L1 by EVs. And this is really, really amazing to me because I studied prostate cancer. And the Cell paper in the beginning, they used some prostate cancer cell lines. And the author found, because it's kind of like a common sense for a lot of prostate cancer researchers, that prostate cancer is a very cold tumor and checkpoint inhibitor doesn't work there. There are no PD-L1 and a lot of things. But the author found actually there is a biology that the cancer cell, they do not have PD-L1 expressed on themselves. However, they actually actively release a lot of EVs carrying PD-L1 and play a function. So this is really cool. And the last but not least is PD-L1 is a promising biomarker for anti-PD-1 or anti-PD-L1 treatment. So back to the current research and what did this paper say? I summarized the kind of like the logic and the different steps the authors did, the experiment. So first, this is the outline, and later I'm gonna go deep one by one. So first is the authors did some experiment on the cell line level, the in vitro study. So they used one non-cancer cell line, which is human lung cancer, human lung fibroblasts, and 3 cancer cell lines, 2 from human, 1 from mice. And then after the cell line experiment, they move on to the patient. They start to test the plasma EV and the PD-L1 expression level on plasma EVs in a cohort of patient. So the cohort, the name is exomel. I think it's like exosome plus melanoma, something like that. And in the beginning, they have 100 patients. And I checked some detail, like 84% of them, the primary melanoma is actually on the skin, is cutaneous. And at the time of inclusion, 74% of them were stage 4, and 80% of them was like M1C to M1D disease. So which indicate these cohort of patient actually is like a lot of other checkpoint inhibitor cohort, they are a patient with late stage cancer. And then after they checked the plasma EV level, like EV expression of PD-L1 level, and then they gave the patient the treatment. Interestingly, later I'm going to present they are not just anti-PD-1 treatment. also include like anti-CTLA-4 treatment and target therapy. It's a mix. It's a mixture. And then after a median time of 4.5 months, and then they test the patient for the second time. However, at that time point, 23 patients died and 31 patients lost follow-up. So eventually only 46 patients in this cohort have data from both time points. This is the outline of the paper, and I'm gonna go into them one by one. So first is experiment in the cell line level. The authors trying to do three things or achieve three goals. The first is PD-L1 expressed in cancer cell or patient-derived EVs. And second, PD-L1 was expressed on the EV membrane or the safe surface. And third, do they have a function? Can EV PD-L1 like mediate T cell immune suppression? So in the beginning, I know like almost everybody in this group are interested in like how they isolate EV, because this is the big factor that may cause a lot of problem or disagreements within the, between studies. So I just listed it here. And here's the EV isolation from the protocol the authors use from cell culture medium, CCM, or plasma. So in the CCM EVs, I did pay attention. So the authors put the cells in the EV depleted medium before they isolate the EV, and the CCM was collected, and then after a bunch of different speed of spinning, like low speed, medium speed, and then they actually depleted the particles palleted in like 10,000 G. So which for me, it sounds like they are studying so-called like small EVs or relatively small EVs. So after they collect the pallet at 100 G and 100 kg, sorry. And then after PBS wash, and they use the EV. So in the plasma EVs, the peripheral blood was collected, and then this is a little bit different from what I think the guidelines suggested. So they spin it for one time at about 2,400 G for 15 minutes, and they store the plasma. I think a lot of people use a low speed to separate plasma from the whole blood, and then spin at this speed for twice, right, to remove the platelets and a lot of other things. So 5 ml of plasma was used for each patient in this study. And after the plasma was thawed, they spin it with another low speed, and then also, again, through the large particles, and then they collected the small particles after 200 kg. This is the protocol they used. So here's the data, and the first they want to prove PD-L1 actually were expressed in EVs from cancer cell lines and cancer patient plasma-derived EVs, but not in non-cancerous cell line or healthy donors plasma. And they try to do that by Western blot, and these are two blots. On the left side is the blot for cell line, and on the right side are blots for plasma EV. So one thing need to be clarified is, like, unfortunately, all the figures in this paper, the quality is very miserable. I don't know why. And so here, the markers the authors used is ALEX, CD63, TSG101. These are all EV markers. And the GRP94, they use it as a EV negative marker. They come from the ER as a contamination. And they use actin as a loading control, and the PD-L1 is their target. Oh, sorry, I missed CD9, another tetraspanning as an EV marker. So as the result, you can see none of them expressed the GRP94 except for the cell lysate. And all the EVs are positive for the EV markers, and only cancer cell line and EV from the cancer patient plasma express PD-L1. Not non-cancer cell line or healthy donors plasma EV. So when I read these figures, I have two, not question, but a little bit surprised. The first is CD63, and in a lot of research, including mine, and they were kind of like a smear. But in this paper, the author used a different clone number, so which is definitely possible. And the acting, so it's hard to find really a good loading control for EV Western blot, as far as I know. But here, I think the author did a really good job. I mean, the acting looks pretty. Oh, here, there's a side note. The blood samples were pooled, actually, for this Western blot from 6 melanoma patients, 6 lung cancer patients, and 5 healthy donors. It is not just from one patient of 5 ml blood. And also did the NTA, nanoparticle tracking analysis, try to compare the EV numbers and the size. So without any doubt, like no difference was found in the total number and size of EVs between cancer cell line and the non-cancer cell line, as well as between the cancer patient and healthy donors. So as here, you can see here, they only put one representative figure of the size distribution here. The particle sizes range from like about, I think, 60 somewhere to about 200. So they were fit in a size of like small EV. And on the right side are the particle counts and the median like size. And they also did some statistics, and there are no significant difference between the size or particle number or concentration. So first, they proved the PD-L1 actually were there, and the second is like, where is it? So they want to do some experiment to check if the PD-L1 is presented on the membrane of the tumor-derived EVs. So to determine the PD-L1 expression is on the EV surface, protein-protein interaction experiments were conducted. To be honest, personally, I have never done this experiment by myself. Sounds like I put the whole paragraph to explain the method, but briefly, sounds like a immunocapture assay. So first you need to coat the PD-1 and on a chip, and then if the PD-L1 is expressed on the surface, EV surface, then they can be captured, and a sensor can give you a reading of the binding. So as the result, the binding curve, as you can see here, so all the cancer EVs, cancer EV from cancer cell or EV from cancer patient plasma, and they do have a higher binding, and the author proved kind of they are on the surface, or a lot of them are on the surface. And then, so they want to prove if the EVs, which carry PD-L1, do they have a function or not? So here they also conduct a very classic immune assay. What they did is they put the EV in a co-culture with T cells. So first, also I learned a little bit from my colleague. I'm not an immunologist, so I asked like, what is this experiment for? So the T cells were enriched by Roc-Curve, which is a kit from the PBMC. So basically they draw the peripheral blood from healthy donors, and then they use a density gradient to separate the PBMC, the peripheral blood monocyte layer, and all the immune cells should be in that layer. And then the Roc-Curve, I think it's the magnetic beads coated with conjugated with some T cell specific markers, and then it can capture T cells from that PBMC. And then the purified T cells, and also they give the number, which is 1 × 10⁵, were activated with the CD2, CD3, or CD28 beads. So in a normal setting, the T cells, they were not activated. Otherwise, the immune system going to be blown. And so the CD2, 3, or 28 are the most common receptors on T cell to activate them. So it seems the author used the beads to activate the T cells, and then incubate them with either human cancer cell or the cancer-derived EVs. Sorry, I apologize here. The 5 should be like upright corner, the same as the 6 here. So they checked different markers, like 3 markers of the of the T cell after incubation with either cancer cell or cancer-derived EVs. They are PD-1, Ki67, which indicate the cell proliferation, and interferon gamma, the T cell function. And then, as you can see here, oh, by the way, the PD-1 was evaluated by flow cytometry, and the Ki67 and interferon gamma, they were assessed by IF. So to be noted, just they were assessed by 2 different assays. And as you can see here, so from the left to the right, the left is non-activated lymphocytes, which, as you can think about, it's like a control. It's not activated, so they should be they should have a low expression level of all these 3 markers, low expression of PD-L1, PD-1, and then so the second one is the activated lymphocytes, and the third. Wait, what is that? Oh, sorry. So they are the lymphocytes with or without, they are the T cells with or without the cancer cell or cancer EVs. So as you can see here, the T cell alone is here. So the activated T cell is here. And then the activated T cells with both cancer cells or cancer EVs, they have actually low expression of these markers, which means their function were actually suppressed. So that's what the authors say. And so the second part of the experiment, so after the author did a little bit experiment in cell line, which actually for me, some of the experiment, they were as a reconfirmation, because a lot of experiment and findings were already proved by the Nature or Cell paper. However, they just want to repeat it and also confirm it first in their experiment setting. And the second step, the author want to move on to the patient cohort. And I already gave a little bit introduction of this cohort. So here the author want to do three things at the first time point. The first is they want to check the plasma EV PD-L1 versus the plasma soluble PD-L1. And the second is plasma PD-L1 versus the tissue PD-L1, which is also something I'm really interested in. And the third is association between the EV PD-L1 at the first time point, which is before treatment, and the other clinical features. So the first, the authors tested the level of EV PD-L1 in EV or the soluble PD-L1 in the plasma. And the method they use is ELISA, and also ELISA were used in the following experiment later. In all the experiment, they evaluate the PD-L1 level on EV. And here, as you can see here from the patient plasma, the EV PD-L1, the expression level, the concentration has a median of like 64.26 like picogram per mil. On the other hand, the soluble PD-L1 is almost like non-detectable. So they tested it only in 30 patients out of 100. I don't know why. But this is way enough to prove like actually there are almost non-detectable PD-L1 soluble as a soluble free like protein in the plasma, but there is actually a high expression of the EV PD-L1 in the plasma. And then they're also trying to prove the relationship, or they want to compare the EV PD-L1 versus the tissue PD-L1. So in tissue, they did IHC, as a lot of other experiments did. So here on the left side is a example. So the PD-L1 negative versus PD-L1 positive. And on the right side are the EV PD-L1 expression versus the tumor. So here again, they picked only 30 patients out of 100. And surprisingly on EV, 100% of patients, which all 30 patients, they do have a positive PD-L1 expression. But on tumor, like only 20 out of 30, which is 67% of them, have a positive PD-L1 expressed on tumor tissue. So for me, two things is a little bit surprised. First, in other studies like Nature or Cell Study or some other paper published, usually not 100% of patients have a positive PD-L1 expressed on their plasma EV. But in this study, all 30 patients do have a positive PD-L1 expressed on their EV, on their plasma EV actually. And the second thing is like it's further like proof or confirmed the hypothesis brought up like by the Cell paper. So previously the Cell paper demonstrated there is a hypothesis like some tumors, some cancer cells, they do not have like PD-L1 expressed on themselves. However, they may have PD-L1 carried in their released EVs. So that is something fascinating to see. And the last but not least to see in this part is also trying to, before they give the patient any treatment, they want to do some analysis to see if the baseline EV PD-L1 level can be associated with any clinical pathologic features. Unfortunately, none of them were related, except for one, which is the decrease in lymphocytes. However, the p-value is about 0.048. I don't think it's really significant. Just by number, it's like less than 0.05. So in general, all those factors like age, gender, and the tumor burden, pathological subtype, and BRAF mutation and stage, metastatic sites, all those things were not related, not associated with the baseline PD-L1 level. So after they gave the patient treatment, and then they move on to the second time point, and also is the end of this, not the end, they actually have another follow-up of 9 months after that, but this is the last part of the experiment. So in this part, they want to demonstrate three things. The first, they want to test association between the EV PD-L1 changes, which is the time point 2 minus time point 1. And they want to explore the relationship between this delta PD-L1 and the treatment response. They want to test the association of this delta PD-L1 and the clinical outcome. And last but not least, they want to see if they can use EV PD-L1 to monitor disease progression. So before I move on to the data, I want to give a little bit explanation in the clinical features of these patients, like 46 patients with both S1 and S2 results. To be honest, I don't know why it's S1, S2. It's like T1, T2, but just 2 time points. S1 is pre-treatment, S2 is post-treatment. So within these 46 patients finally stayed and have both like data points. So 36 of them received anti-PD-1 therapy. 2 patients received anti-CTLA-4 treatment. 8 patients received anti-BRAF1, anti-BRAF and anti-MEK targeted therapy. So as I mentioned in the beginning, it's not purely a cohort for checkpoint inhibitor or purely for PD-L1, for PD-1 therapy. It's a mixture. But all those patients received like non-PD-1 therapy, they actually were treated with anti-PD-1 therapy before. So they call it like associated with anti-PD-1 therapy. And among all those patients, 5 patients will experience like complete response. We call it CR. 23 patients experience partial response. 8 patients experience stable disease, no change, and 10 patients unfortunately experience progressive disease. And to be noted, actually, as I presented before, a number of patients already lost follow-up or died. And here, the patient left. In the end, 10 patients like experienced PD. And I also put a star there is because interestingly, these numbers, 5, 23, 8, and 10, these numbers reported in the method part is actually different from the number reported later in the results. I don't know why, there probably is a typo there, but slightly different. So this is the result, and as I mentioned, they're also trying to first see if the delta PD-L1, the changes, was associated with like disease status, like responders versus non-responders. So first, from the upper left, the figure here, the author did, so the black bar is the PD-L1 expression on EV, like before treatment. The gray here is the post-treatment PD-L1 expression on EV. And in the responders, there are no significant difference observed. However, you can see actually there is a decrease, which means the expression of PD-L1 on EV decreased a little bit in responders. However, on the other hand, in non-responders, so in the patient with a PD, progressive disease, the second time point actually have a way higher EV PD-L1 expression level. So as you can see here, before treatment, it was like about 80 something. And after treatment, it's like 350. So the difference is significant. And here on the upper right, the figure, the author separate the patient into 2 groups, responders versus non-responders. And if you check their median delta PD-L1, as you can see here, the delta PD-L1 is significantly higher in the group of like in the group of non-responders. So which means after treatment, if a patient does not respond to the disease, and then its plasma EV PD-L1 level gonna increase. So this is the figure gonna tell you. And here, the waterfall plot, as you can see here, so on the x-axis is the delta PD-L1, and if it's like negative, the delta is negative, means the PD-L1 level actually decreased. And if it's positive, means it's actually increased after we give the treatment. And and black color is the responder, and gray is non-responder. And as you can see here, all the patients who have a negative delta, which means whose PD-L1 level on EV actually decreased during the treatment, and all those patients were responders. And within the like the patients who have an increased PD-L1 level, there are mixed of responders and non-responders. And here on the right side, this figure is a little bit misleading. So I spent a little bit of time to fix this, to understand this better. And here, what they want to say is, in responders, like 100% of the responders, they actually have a negative delta, which is actually the same as presented here. But as the delta positive, there are 66% are responders, there are 100% patient that progression. So they actually combine the gray bar with the black bar. That's what I think. So what they're trying to say is like the responders, so they all have a negative delta. However, with a positive delta, it's 100% progressed. However, within them, there are still some responders. Does that make sense? So later we can raise questions, but they're trying to say is like with the positive delta, which means the PD-L1 increased, 100% of them actually have disease progression, but there's still a percent, like 60% of them, they're responders. So this was a table tell me, a little bit confused. So then this curve is the most confusing part in this paper for me in the beginning. So as a lot of biomarker research trying to do, the author used ROC curve to analyze, to trying to set a cutoff and to see the accuracy of this biomarker. So first of all, what I'm trying to see is what I'm trying to say is like I think there is a mislabel in this figure. Usually it's 1 minus specificity. It's not specificity. So how does this curve work? As just in case some of you are not familiar with ROC curve, is they actually generated all the data. So they tested each delta number as a cutoff. And under each cutoff, they're trying to see the sensitivity and the specificity of this test. And then they build this curve. This is the ROC. And on the curve, you can actually try to use different methods to locate a spot, to locate a point, to choose it as your cutoff. And usually people choose the point as to make both sensitivity and specificity as high as enough. However, in this curve, as you can see here, I think there's a mislabel. If this is correct, then at this point, the specificity is 100% and also have a super high sensitivity. This is not correct. So first of all, on the right side, but the data is correct. I just say there is probably a mislabel there. So the AUC, which is area under the curve, is 0.867. And the AUC usually directly indicate to the accuracy of this biomarker for diagnosis. And the 1 is the best, and anything below like 0.5 is like very bad. So 0.867 is relatively good. And the p-value is less, is very significant. And the optimal cutoff based on the ROC curve the author picked is 100. So what they're trying to say is like, when the delta, the change of PD-L1 is more than 100 picogram like per ml, then they think it will have the best sensitivity and the specificity, the best balance of these two. So under that cutoff, and you can see the sensitivity is 80% and the specificity is 70%. And the positive predictive value is 91%, which indicate 91% of patients who has a positive diagnosis, which actually have a true positive result. And the negative predictive value is 54%, which indicate 54% of the patient who has a negative diagnostic results actually really have a negative results. So this is how these data mean. So if you just check the curve, you will see all these numbers are not bad, and they successfully set a diagnostic like cutoff. However, two things. The first is, as I said, there's a mislabel. The second is, as you can see the curve, usually if you really have enough patients in the cohort, you will have a lot of data sets, and then the curve will be way more smooth. But here, in order to make this curve, the author, there are only like 64 patients, 46 patients they can use. You can see it's not a very smooth curve, and they don't have, I don't think they have like enough patients to make a really smooth curve, like in ROC. So let's move on. The most important part is about the prediction. So the author already demonstrated the baseline PD-L1 actually not related to any of those clinical features. So they want to see if the delta, the PD-L1 change, can predict the survival or not, because they already know the responder, non-responder. There's like no way to use the change to predict the responder, non-responder. They can only see if there are a significant difference between responder and non-responder. However, after the end of after S2, after you can see the responder and the responder, so you can still like keep follow up, and then you have a survival like information. So here the author did a bunch of like analysis and pseudo regression, the multivariate analysis. The author found the delta PD-L1 was an independent predictor for overall survival, the OS. I red box highlighted it. So it's a mixed feeling here. So first, I list some information here. The median follow-up was 9.9 months. And also the author indicated in univariate analysis, the baseline PD-L1 level actually were not associated with OS and PFS. It's not in the table. However, they mentioned if you only use the pre-treatment PD-L1 level, you cannot predict a survival. But as the table showed here, if you use the delta, the PD-L1 change, you can successfully predict the OS. It seems to be like that. However, the p-value is significant, but if you look at the HR, the hazard ratio, it's just 1.004. So what does hazard ratio mean? So again, I'm not an expert of statistics, but usually people take this as like, you can evaluate the risk. Like, for example, if the hazard ratio, the HR is 4 there, then it means if the patient, like the change, the delta PD-L1 is more than 100, then it have 4 times of risk, like to have like to die or to have like a shorter like overall survival, something like that. However, the difference, however, the HR here is only 1.004, which is actually which is so-called statistically it's significant. However, in the real practice, it's almost the same. And also the author put a star there, and I read in the figure legend, I found the star means the primary cutaneous melanoma patients, which means, I guess, in order to get this p-value and just this item, probably it's not conducted in all 46 patients. It's only in a subgroup of patients, which is 36. They have primary cutaneous melanoma. And then they also did a similar analysis, and they found the change, the delta PD-L1 was also an independent predictor for progression-free survival, just another parameter of survival. And here they also use this cutoff to separate the patient cohort to 2, which the one is, the first one is the delta is less than 100. The second group is the delta is more than 100. And of course, the delta is more than 100 means there's an increase in the PD-L1 level, which is the yellow curve here. And after they separate the 2 groups of patients, they found there is actually a significant difference in their OS and the PFS. So on the left side is the progression-free survival. So for the patient group who has a delta PD-L1 less than 100, supposed to be the responders, the median PFS was not reached, which means actually not reached 50%. The median means like the 50% of patients who have progressed. However, at the end of this follow-up period, like they did not reach to 50%. There are less than 50% patients who get a progression. However, in the PD-L1 delta, more than 100, which is supposed to be the non-responders, and the PFS is 9 months, which means within 9 months, about half, like 50% of this group of patients who has a progression. And the p-value is 0.100, which is significant. And on the right side is the OS. And as you can see here, the median not reached for both groups, which means the overall survival, like none of the group of patients have like 50% of patients died. So, well, there are different ways to interpret the Kaplan-Meier survival curve. So as here, I'm not a medical oncologist, I'm not a dermatologist. I have no idea like what usually in those checkpoint inhibitor clinical trials, how the survival curve gonna look like. But as you can see here, one thing I'm pretty sure is like the 2 curves, they separate very clearly in the beginning, which means it's not like in the beginning they're the same and in the long term they're different. It's in the beginning, these 2 curves, these 2 groups, the progression rate is very different. So at last but not least, the authors trying to see if they can use EV-PD-L1 to monitor the disease progression. And I don't know why, probably they don't have enough data, but in this section, they only include 8 patients out of 46. So the data is only like, it's a case study. It's not even with any statistics. So, but what they find here are some individual cases. And as you can see here, the curve indicates the level of EV PD-L1 level. And here, down there are the imaging, indicates the disease is progressed or not. What they found is during if the tumor actually responds, there's a decrease in tumor size on imaging. Then there's also a correlated decrease in the EV PD-L1 level they can detect it. And at the same thing here. So if the tumor size increased a little bit in the beginning, the time point 2, the PD-L1 level also increased. And then after a while, the time point 3, the tumor size actually decreased. And then the PD-L1 level in the plasma EV also decreased. So they tested like head and neck lesions. They tested like on the lesion on the lymph node, the leg, and also in the brain. All the same thing. In these 4 patients, they found actually the PD-L1 level perfectly correlate with disease progression or regression. So, however, for me, there are 2 points. The first, there's a side note here. The author mentioned this actually, the PD-L1 prediction did not only work for anti-PD-1 therapy, but also targeted therapy, which means, for example, this patient, actually, he or she did not receive anti-PD-1 treatment. It could be like anti-BRAF, like targeted therapy. But if you evaluate the PD-L1 level on the EV in plasma, it's still like correlated with the disease progression or regression. This is very interesting. And for me, another point is, again, I'm not a medical oncologist, but I do know there's some idea like a pseudo progression. What does that mean? It's like, as the medical oncologist observed, so especially a certain group of patients who receive checkpoint inhibitor or other immune therapy. So after they give the treatment, and if you just look at the imaging scan, there is a period of time, probably the size gonna increase a little bit in the beginning. And then the size of the tumor gonna keep decreasing. And they call it like pseudo progression. And there are a lot of explanation and research going on in that field. I'm not an expert, but one commonly accepted thinking or hypothesis is, in the beginning, the pseudo progression is not real progression of tumor. Cancer cells did not really grow. It's actually the immune reaction. So if you look at the curve, it's not, in this part, I also did not do any statistics. There are only 8 patients, but I just have a concern. For example, for immune therapy, if there is pseudo progression, actually the tumor burden, real tumor burden actually decreased. However, on the imaging, the tumor size actually increased in the beginning. How the PD-L1 gonna tell us? I have no idea. So here's the conclusion of this paper. So the author presented the study. The presented study offers the rationale for using the EV PD-L1 as a predictor of treatment response in melanoma patients, with the advantage of its non-invasive or minimally invasive collection and real-time monitoring. And also in this work, the part of the first conclusion has already been proved by other paper too. But in this work, the authors further demonstrated in a larger and prospective cohort that EV PD-L1 may be a marker of different therapies. So it's not just for immune therapy. And the last but not least, the author also mentioned there is a multi-center clinical study with melanoma patients ongoing to establish a more precise cutoff values. So for me, the last slides, I listed some of my questions and thoughts for the future. First, the TM was not presented at all time, which is something disagree with the MySEF recommendation. And the second, I do have a question like, can baseline EV PD-L1 predict response at all? The author actually demonstrated the baseline PD-L1 is not associated with any other clinical features, and the baseline PD-L1 cannot predict survival. I'm not sure if the author is trying to say that equal to like cannot predict response. And the third is the predictive value of EV PD-L1 really needs to be further explored. Because for me, from this paper, besides the conclusion the author made, I have some personal conclusion. The first, of course, is very valuable. The author proved there is a difference of EV expression of PD-L1 versus the tissue PD-L1 expression. That is really fascinating. However, we need to think about if that's true, what does that imply? So the author first proved, like I mentioned, the baseline PD-L1 cannot predict the survival. And then even the delta PD-L1, the author did the only, I think the positive data is the author demonstrated the delta can predict the survival. However, just like I mentioned, the HR is almost like 1. So I don't think it's really significant, though it's statistically significant. So after all these, I did not see really a predictive value of EV-PD-L1 or the delta EV-PD-L1 here. So it sounds like more like the only thing the authors showed after all these is like the PD-L1, they can use it to monitor the disease progression according to the imaging, but without further exploration. But for me, it sounds like it's nothing to do really to predict immune status. It's like something related to tumor burden. Because if the diseases keep like progress, then the EV-PD-L1s keep increasing. So that's something has been proved here. That's why, after all, I think the predictive value of EV-PD-L1 really needs to be further explored. And the fourth point is circulating EV-PD-L1 level stable in the same disease phase? That's also a key question from me. The author presented there is a difference between like the pre and post treatment. However, I'm not sure if it's stable if you test the patient circulating PD-L1 on EV at the same time point. At least the triplicated experiment were not presented here. So the fifth point is like where it'd be more efficient and reliable to isolate EV from tumor tissue. So several years ago, I read a paper from Dr. Yang published on Journal of EV, and that's the first paper I saw, like trying to establish a protocol to isolate EV from fresh tumor tissue. However, in this study, it would be very interesting to see, because though the author demonstrated there are a detectable PD-L1 on the circulating EVs, but a lot of other paper published like actually presented the true EV presented in plasma, even isolated by UC, is relatively low. So there still could be a lot of contaminants, and within those true EVs, it's from circulation, and any cell type in the body can contribute to it. And there's an interesting paper calculated how many EV exactly really from the cancer, a piece of fingernail tumor tissue can be found in like 5 ml plasma. So actually, if we do the calculation, if the calculation is right, then there's very low chance to find really cancer-specific EV in the plasma, but theoretically. However, I mean, if we get a piece of tissue and we directly get EV from that, just thinking, probably it could be more efficient and reliable. And the second point is the single EV analysis and EV subpopulation analysis will be very helpful because the PD-L1 were not only expressed on cancer cells, it could also be expressed on immune cells like macrophages and dendritic cells, and also some other cell types to play a role. And it will be interesting to see the co-localization of some other cell-specific markers. And the last but not least is like, how about use this with other markers together? On the right side, this is the last slide. So on the right side is a figure I pulled from a Nature review. So these are all 3 most important things I also think to predict or affect the response of checkpoint inhibitor biologically. So it's not just PD-L1 expression, also related to mutation burden, the tumor infiltrated T cells, all those things. So eventually it may not be just one player there. So I would be very willing to see in the future if there are any other study like actually combine the PD-L1 expressed on EV with some other tools and to make a better prediction. So that's everything I have. Thanks a lot.
Thank you so much, Liang. I don't know if you did pay attention to the chat box, but Ken is experiencing some Wi-Fi issue and microphone issue. So we're going to lead the discussion part together with Dominic. So first of all, thank you so much for your nice presentation. We do have a lot of questions in the chat box. So we're going to start with some naive questions. So let's unmute Jayanta Dibnath, if any co-host can help me on that.
Yeah, good to go.
Okay.
Hi, can you hear me?
Yeah.
Yes, we can. Go ahead.
Hi. Oh, thank you very much for the presentation. I had two quick questions, both technical or you know, just pretty detailed. The first was you showed that nice review showing that there's patients that were responsive to therapy, but they had differing levels of PD-L1 expression.
Yes.
And so one of the questions I had is actually, especially given the fact that it looks like 100% of these are coming out in EVs, there are a lot of different PD-L1 antibodies out there. And, you know, I know this was kind of a meta-analysis, but did it take into account that this could just be differences technically in terms of staining?
Yeah, that's a very, very good question. So in this, do you mean this paper?
Yeah, this one, yeah.
Yeah. So there is actually larger review in Nature, and this is not a Nature review. This is a reference of that. I just want to use this figure. However, both paper they actually take into account the clone of antibody, and not just the antibody they use, but also the threshold, the diagnostic, the standard they use. Because what is a positive for PD-L1? Some research said it's 5%, some research said it's 1%, some research said it's like 15%, and that's also different. And the way they stain it is different. And also they expressed on the cell type. Some research, it has to be like on cancer cells, but some research doesn't care. So that's a very good point. So in this field, in this review specifically, they actually did focus on that question.
And very quick, I think someone asked it a little bit later, but it looks like the trend in PD-L1 circulating expression is a bit different from what was described in the Nature paper.
Exactly, yeah.
And I don't know what your thoughts were on that.
Yeah, so I think first of all, I think both of the cohort are kind of like melanoma patients, but I forgot exactly the cohort size of the Nature paper. I'm not sure if it's larger or smaller. So, but in this one, the author also mentioned it's very different compared to that one.
Let's to cover this question unmute as well some folks here that were very active in the chat box.
Let's get Fabrice in the mix. Let's see if Brian is still here.
Yeah.
Okay, Brian, you're also unmuted. Feel free to pitch in.
Thanks, Dominic. Well, I was watching this with great interest, and wonderful job, Liang.
Thank you very
great
much.
Presentation of the paper. Sorry, my dog is barking. Well, I thought that a lot of the comments in the group chat were excellent, and I would certainly reiterate some of those same points. I thought Fabrice made a good point that as a clinician looking at this, a clinician scientist, I look at this and I do think that it's pretty far from translation to the clinic so far, but we don't have to be there yet. It takes time. And I think, you know, some of the things I'm wondering about is I think there's a lot of small melanomas that get resected, and the expectation is those people are cured and nothing happens to them thereafter. And I don't know, you know, when do you have sufficient tumor burden? This is a little bit related to what Genevieve was asking a moment ago. You know, at what point would you have enough melanoma burden that you could actually begin to see any signal in the plasma? And I think the questions about the appearance of the signal and the disappearance, the dynamics of that are incredibly important, but I don't currently know of a way to study those things in humans.
Yeah, thank you very much. Yeah.
Some observations. I'm not sure it's a question exactly, but you may have some good thoughts about those things.
Yeah, thank you very much. I think it's a very good point. I think, yeah, I totally agree. There is still a distance between from now and to really like clinical application. First is this paper and a lot of other papers, the authors trying to explore the EVs and the PD-L1 expression on EVs from the plasma. And Dr. Whitwer group and And our group, we actually collaborated and did a research trying to check the quality and the efficiency of EV isolation of different isolation methods, and also most importantly, in different sample types, and in urine and plasma and other things. Actually, plasma is really a challenging sample to work with, and I know it's really fascinating because it's easy to get, it's draw blood, but however, there are a lot of contaminants, so can actually affect the efficiency of your target marker check. And also, on the other hand, I just listed in the end, one of my thoughts is like, I do think probably if, it depends on the purpose. One way to do this is like, I would be really interested to see if you get a piece of tissue, no matter how big it is. It could be metastasis, it could be primary tumor, even on melanoma, I mean, lung cancer, or like in my field, like bladder cancer, if we get a piece of tumor freshly. And then we get, we do have established protocol, several paper has already been published, and including our group, like to get EV from those fresh tissue. And then I think by doing that, probably we can have like more directly a direct talk with EV from specifically from that tissue. Though again, those EVs can still come from different cell types. It could come from the immune cells in that piece of tissue. It could come from the cancer cells in that piece of tissue. But if you really want to see the cancer feature, how it's going to predict the response of a certain treatment, I just think probably it could be more efficient compared to if you try to find a needle in an ocean of plasma. Yeah.
Thank you, Liang. So then we had also a question from, let's see, Ranta, regarding the cleavage of PD-L1 in EVs. Ranta, are you here?
Yes, hi, I am here.
Please ask your question.
I wanted to ask because I've heard from some teams that there might be some proteins that are cleaved in the vesicles, and if you ever encountered this particular protein to be cleaved in the vesicles, which might lead to different effects than the whole protein.
Sorry, you mean some protein could be cleaved on the EV?
Yes, or as a cargo, it can be cleaved by different enzymes in the cells, and in the vesicles they are cleaved. So naturally, they will have different effects.
Yeah, that's a great thought. I mean, definitely, I think that's going to affect a lot of biology. But however, I personally don't have any experience or knowledge in this field. Can any other colleagues may have any thoughts? Because in this paper, they did not really dive into the biology, and they just get a sample, and if you see it, you see it. However, it's a great question. It's a great question. Like, are there any other factors probably going to hide it, or yeah, I don't know. But comparing to the paper from the Nature, the previous question, the previous paper in Nature, like they did not see such a high percentage, which is 100% a positive rate of PD-L1 on EV. And why is that? Well, at least they use the same isolation method. If this paper did not use that, that could be an obvious reason, but I think both of them, they use UC. Then I don't know, but probably there could be some biology there. Yeah, I don't know.
Okay, thank you.
But then, let's see.
Let's jump to the next one.
Taral have a question as well. So Taral, I'm going to unmute you. Are you here, Taral?
Yeah,
Okay.
yeah, I'm here. Can you hear me?
Yeah.
Yes.
Yeah, my question was very simple about why the authors use different centrifugation protocol for plasma and for cell culture supernatants. Did they mention or did they point out something on that?
So first of all, no, they also did not mention the reason why. But on the other hand, according to some position paper, or I'm not sure if it's in the myself, so the isolation protocol by ultracentrifuge for CCM and the plasma, they are different because they do have different components in the beginning. there are way more small vesicles, and the vesicles share the similar density as EVs in plasma. So it is different. As I think most commonly used protocol for CCM is exactly the one that also used here, but for plasma, it's a little bit different. Usually they separate the plasma first, and then they spin with a like 2500 G for 15 minutes twice. That is a different step, like the CCM do not have, and that step is specifically designed to exclude those like like the, I think, platelets or some other fibroblasts, something like that. And then it's the same, which is a high speed if you want to remove large vesicles. And then it's again the same. They use ultracentrifuge, which is from 100 kg to 200 kg to palette the EV. However, I do notice here the author used 100 kg to palette CCM EV, but 200 kg to palette plasma EV. And I checked the rotor, not the rotor, I mean the rotor. I think the information, at least according to limited information provided in the paper, they are the same. So I don't know why.
All right, thank you everyone for your input. Again, Dr. Whitwer apologized for missing this very nice discussion, but unfortunately he's dealing with some IT issues. Thank you again for joining, and please stay tuned. We're going to resume next week with another presentation. Bye now.
Thank you very much.
Thank you.
Bye, everyone.
Thank you.